Good morning, students. Welcome to the world of SciTech, Science and Technology. Today we are discussing about DNA and chromosomes. What is the relationship between DNA and chromosomes? DNA is deoxyribonucleic acid. It is composed of 4 deoxyribonucleotides, that is deoxyadenylate, deoxyguanylate, deoxycytidylate, and deoxythymidylate. What is nucleotide? DNA is made up of a chemical building blocks called nucleotide. This chemical building block is made up of 3 parts: a phosphate group, a sugar group, and 4 nitrogen bases. Sugar group is deoxyribose in DNA and ribose in RNA. Phosphate group is phosphoric acid, H3PO4. Phosphate group react with the sugar in nucleotide to form an ester bond. The link between the nucleotides are called phosphodiester bonds. Each base pair is joined by hydrogen bonds. And the base sequences are, the nitrogen bases are adenine, guanine, cytosine and thymine in DNA. Adenine, guanine, cytosine, uracil instead of thymine in RNA. So that we can conclude DNA and RNA are composed of nucleotide that are linked to one another in a chain by a chemical bonds between the sugar base of one nucleotide and the phosphate of adjacent nucleotide. Sugar is in 3' end and phosphate is in 5' end of each nucleotide. The deoxyribose and phosphodiester bonds are same in each nucleotide. Next we move on to genetic information. The coding. The sequence of genetic information are coded in the sequence of bases. I repeat, sequence of bases. What are the bases? Thymine, adenine, cytosine and guanine. So that the basis is altered. If the basis is altered, the genetic information is altered. The sequence of bases portion in DNA are called gene. Gene code contain or DNA code contain an instruction to make a protein, and a molecule is essential for growth, development and health. We know that DNA is a right-handed double helical structure. Each strand runs in opposite direction to one another, that is anti-parallel. One runs in 3' to 5' order and other ones runs in 5' to 3'. That is called sense and antisense. These two strands are separated by DNA replication. Human genome is made by about 3.2 billion bases of DNA molecule. Okay students, we move on to what is the relationship between DNA and chromosomes. So let's move on to that topic. We know that DNA is a double-stranded molecule and while RNA is a single-stranded molecule. DNA is stable under alkaline conditions while RNA is not stable. RNA convert the information stored in DNA into proteins. We know that the genetic information stored in DNA as base sequences. So that this information stored in DNA into proteins. mRNA carry the protein blueprint from a cell's DNA to its ribosomes, which are the machines that driven protein synthesis. The DNA molecule is packed into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structures. We discuss about later. Polarity of DNA molecule. What is it? In the case of DNA, The base sequence is always written in the form of 5' end to the 3' end. This is called the polarity of DNA chain. Purines and pyrimidines. What it is? These are the nitrogen bases that hold DNA strands together through hydrogen bonds. The purines in DNA are adenine and guanine, the same as in RNA. The pyrimidines in DNA are cytosine and thymine. In RNA, they are cytosine and uracil. Denaturation of DNA strands. When a DNA solution is heated enough, the double-stranded DNA unwinds and the hydrogen bonds that hold the two strands together weaken and finally break. The process of breaking double-stranded DNA into single strand is known as DNA denaturation. At the lower temperature, the melted strands are reassociated. This is called annealing. The top three methods of DNA denaturation are heat, sodium hydroxide treatment, and salt. Each of these methods will break the bonds between strands. If a protein in a living cell are denatured, this results disruption of cell activity and possibly cell death. They are especially beneficial to bodybuilders who use denatured proteins. They help aid in protein absorbance and protein digestion since the protein is already broken. With eggs, this happens most frequently when they are heated or beaten. Histones, they are protein containing higher concentration of amino acids. These are five classes: H1A, H2A, H2B, H3, and H4. Histones are synthesized in cytoplasm and migrate to the nucleus. Histones bind to DNA, help give chromosomes their shape, and help control the activity of genes. Most of DNA is found inside the nucleus of a cell where it forms the chromosome. So that we can conclude a single length of DNA is wrapped many times around lots of proteins called histones to form structures called nucleosomes. These nucleosomes then coil up tightly to create chromatin loops. These chromatin loops are then wrapped around each other to make a full chromosome. What chromosomes make a male or a female? Males have XY sex chromosomes, while females have XX sex chromosome. The male can contribute the X or Y chromosome, while the female must contribute one of their X chromosome. Chromosome 1 has 2968 genes. The first 22 pairs of chromosomes are called autosomes. The 23rd pair of chromosomes are known as the sex chromosomes because they decided if you will be born male or female. In humans, each cell normally contains 23 pairs of chromosomes. Diploid number is 46 and haploid number is 23. That means every cell in the human body contains 23 pairs of such chromosomes. Most DNA is located in the cell nucleus. It is called nuclear DNA. Small amount of DNA can also be found in mitochondria. It is called mtDNA. During cell division, chromosomes first replicate so that each daughter cell receives a complete set of chromosomes. Following DNA replication, a chromosome consists of two identical structures called sister chromatids, which are joined at the centromere. Chromosomes have generally three different shapes: rod shape, J shape, and V shape. These shapes are observed when the centromere occupies terminal, subterminal, and medium position on the chromosomes respectively. Chromosome size is measured with the help of micrometer and mitotic metaphase. The supercoiled DNA with histones. These are called nucleosomes. We learned about it. Will have a diameter of about 9 nanometer. A group of such nucleosomes form the DNA fibrils. About 6 such fibrils are further supercoiled to form 30 nm diameter chromatin fibers or chromatin threads. By this time, DNA is folded to about 50 times. Histones stabilize these fibers. These fibers are further supercoiled and condensed to form chromosomes during the metaphase of cell cycle. The packaging of nucleoproteins within chromosome is specific depending on the length of chromosome and the position of the centromere. The chromosomes are numbered. In human, there are 23 pairs of chromosomes. So that from our study, what we understood, a chromosome is a long chain of DNA molecule that contains part of all the genetic material of an organism. DNA is a fundamental molecule that carries the genetic instruction of all living organisms. DNA is packed into chromosome with the help of special proteins called histones. One chromosome has two strands of DNA in a double helix. Students, it is enough today. In the next class, we will discuss about RNA. So, we will meet again in the next class. Bye, children. God bless you.